the second day of the EntheoGeneration Psychedelic Lecture Series. We are thrilled to have Bill Linton, Pancho Meisenheimer, and Alex Sherwood of the Usona Institute, who are going to talk to us about the FDA approval process and beyond for psilocybin-assisted therapy. So can we all have a hand?
Okay. Ron, thank you so much. Really appreciate what you've done here to help put this together. I'm really just going to make some introductions and some recognitions and then kind of set the stage for Usona. These are some of the guys that are doing the most phenomenal work, particularly on the chemistry, and we have a whole team of people led by Laura McCormick, Malia Nutzinger, many others who are just doing incredible work back in the home base in Madison, Wisconsin. I really want to give an appreciation to David Bronner and his team for what they did and the amount of time that they did it to put this together. The design team, the construction team did an incredible job. And Andy, I just saw you a second ago. Thanks so much for, yeah, there we go. Thanks so much for what you did put together this program. Thank you. So we actually have several members of the Usona team here. You've already met Alex Sherwood, one of our chemists, Pancho Meisenheimer, who runs chemistry as well. Christy Kalo. Christy, I want you to raise your hand. She's one of our chemists as well. There you are. Thank you. Another chemist, we have Robert Carbo. He's in San Luis Obispo, California, working at the bench as we speak. We like that. And Troy Good, who's a chemist and project manager, here's in the room as well. Rosmi Kars, research and information coordinator. Rosmi, where are you? Troy, where are you? Woo, thank you guys, thank you. So Usona Institute is a newbie. I mean, we just kind of appeared on the scene about 5 years ago because what was needed at that point in time was to build on the incredible work that had been done in many institutions, New York University, Johns Hopkins, UCLA, some work that had been done at the University of Zurich in Switzerland. And what needed to be done was to put together a team of people with funding to take this through the FDA process. And we're starting with the FDA and then moving to the EMA in Europe. In 2014, we just thought, well, how hard could that be? Oh my, I have to tell you. Presently, we have a staff of 20 people, and we're supported by another 45 organizations and individuals throughout the world that are helping us achieve this goal of getting psilocybin approved as a therapeutic process, as a therapy, psilocybin therapy. It's not just the drug, it's the entire therapy that goes along with that makes that effective. Thank you. So not only does our work rest on the shoulders of people who've been doing this for decades, but I also want to recognize that there are individuals, there are communities, there are tribes that have been working with these plant medicines for thousands of years. Gordon Wasson in 1957 published in Life magazine his discovery of what Marina Sabina and the groups in Mexico had been doing for literally thousands of years and brought it to the West. But our work really rests on their discoveries and what they learned and how they practiced for so many millennia. And I also want to say I've got so much gratitude to each of you for being here. The question was, what can you do? Your presence here is a big step in that direction. Simply by being here and listening to what we have to say, you can go and you will be a ripple effect in the world by taking the knowledge of what you hear and what you learn and bringing that to your friends, to your families, to your schoolmates, to your co-workers, and talk more about what's coming in the world, and it's coming very rapidly. So now I'm going to talk about what we do, how we're doing it, and a little bit about why we're doing it. Hey, it's kind of working. It's pretty cool. Okay, so of course, all humans have the desire to overcome suffering and to find happiness, Dalai Lama. There are millions, hundreds of millions of people in the world that suffer from depression and anxiety and all of the diseases that are associated with this. It's the second largest cause of problems and illness that are in the world. The treatments that we have that was mentioned in the last talk are simply inadequate to handle these disease states with an enormous economic burden. And the burden goes beyond just economies. It touches probably almost everyone in this room in some way. Your family, your friends. It causes addiction. It causes many, many other diseases, eating disorders and things of that sort. All have a common root cause in a break in who we are, a break in understanding the nature of who we are and connecting with ourselves. And these medicines can help in that search. So this therapy is really intended to bring a different view of treatment, to get away from the daily regimen of taking antidepressants. And the discoveries that have occurred in the last couple of decades show us clearly that there's an enormous effect when someone takes a single dose of psilocybin, 25 milligrams. The material is produced synthetically because that's about the only way that the FDA is going to prove it. We've had to make some compromises in terms of, we all like natural products, we like organic products, we like holistic approaches, but the FDA sees the world in a particular way, and if we're going to make a progress in getting this into our society and into our institutions, we've got to follow the breadcrumbs that they put in place to say, this is what you have to do in order to get approval. So what is our vision? The molecule psilocybin is known to be safe. On a weight basis, it's 200 times safer than aspirin. It's effective, as seen in the outcomes of literally hundreds of people. It's something that itself is affordable, and Usona Institute has made a commitment to provide this material at no cost to every research institute in the world for at least the next 5 years. And by the way, we can only do it because of the incredible generous support. The government doesn't fund this. This is through the incredible support of people who have stepped up and provided us with funding to make this possible. And I'm looking at one guy right here who's really helped. Thank you. Thank you, bro. And accessibility is a big thing. We're going to be talking a little bit more about accessibility. What a challenge that's going to be. Okay, so the roadmap, where are we and where are we at with respect to today and in the future? So by the end of September, we're going to launch our next major clinical study. There'll be 80 to 100 volunteers, half of whom will get a placebo, niacin, the other half will get 25 milligrams of psilocybin. And it's on the basis of the outcome of that trial that we'll then take on the next step, which we believe will be the next significant step towards approval. And here we're looking at about 2022, 2023 that we hope to have approval in the United States, and then also in parallel working to get this approved in Europe for treatment there. So a couple of remarks about Usona itself. First of all, we're a nonprofit. This means that we rely on the support of dozens and dozens of people, institutes, and foundations to help us in the studies that are currently underway. We believe in open science. And this means that when we make a discovery, when we invent something, we publish as quickly as we can and we get this into the domain. This means that we do not reach out and try to patent and own and control through patents. This means that discoveries, and Alex can talk about some of the discoveries we've made, and the early publications on synthesis that have really transformed how even psilocybin itself is made. I mentioned also about the providing the material, and then, and we have lots and lots of people that are helping us in our efforts. And I just want to mention just a few of these. MAPS has been an enormous supporter, and Rick, thanks so much for your leadership in providing so much assistance and guidance to our team. We work with the Beckley Foundation. We work with the Door Research Institute in Brazil. Okay. This too shall pass. Imperial College in England, University of Zurich, I mentioned them, University of Jena, and these are all significant research institutes that are looking not only at what these molecules are, but also how they work through brain imaging and beginning to do that very difficult work of even understanding the relationship again between structure and outcome. Okay, so what have we done? So the FDA has granted us clearance to proceed. We really like that. That took a lot of work. We have something called an investigator's brochure, which is a document that provides guidance on how this molecule can be used in research, and we make that available on our website. People can download that at no cost. Let's see, we have seven clinical trial sites that we now have under contract, 5 research universities and 2 contract organizations that are going to be doing the work. Again, think of 80 to 100 divided by 7, and we hope to have this wrapped up in 12 months. So hopefully next year at this time, we can report back to you that this big study has been completed. That's really our goal. Let's see what else is important here. Let's find. Oh yeah, get this. So we can only sign up 80 to 100, but we have 5,000 people already signed up who want to participate in this trial. I don't think we're going to have a problem with recruiting. That's really good news. Okay, so what's next? The expanded clinical facility treatment program is something that we have under design. We also have a number of molecules, novel molecules that we're exploring. The chemists can talk more about that. And again, we're looking at how do we continue to build on what we've done to this moment, and we're looking at creating a world-class research center, probably based in Madison, Wisconsin, that's going to continue the clinical research, the design of new molecules, training and education, which is so important. Okay, clinical research, drug development, regulatory affairs, lots of people involved in all of that stuff. And then finally, I want to mention Chuck Rizon, who's our director of clinical research. And I just want to read what he says. If the large and rigorous studies required by approval by the FDA confirm findings from studies to date, psilocybin will transform how we treat depression, not just in patients who have failed treatments, but in patients who may choose psilocybin therapy early in their disease. So I wanted to read that because the leadership that we have not only has the practical experience, but they have a vision to succeed, and they have the will to succeed, and we will succeed with your help. Thank you.
All right, thank you, Bill. So I want to kind of take you through what it's like to create a drug in a commercially sponsored FDA program. Even though Usona and MAPS are nonprofits, the FDA holds us to a requirement of a commercial sponsor. And those regulatory requirements are significantly higher for drug development than they are for some of the academic sponsorships that we've had. And so let's kind of go through how we kind of go about this. I want to start with a classical drug discovery pipeline, all right? And this is a continuum from left to right. Early on the left side is discovery. Pharmaceutical companies take hundreds of thousands of compounds, maybe millions of compounds, and they screen them in one of 2 ways. One way may be a phenotypic screen, which means that they're just looking for an effect in an animal or a cell or something. Or another way might be a target-based screen. So they know something about the target they're going for, and they just look for a modulator. So they screen in one of those 2 formats, phenotypic or target-based, and they look for hits. And once they find those hits, they might dial down from hundreds of thousands of molecules to hundreds of molecules. And from there, the chemists start to tweak the molecules a little bit, look for some lead optimization. Think Alexander Shulgin twisting the edges of a phenethylamine or a tryptamine. And then from there, once they get down to a molecule, they've got their lead, and they start down the program of early preclinical safety, clinical safety, phase 1, early efficacy in phase 2, and pivotal efficacy in phase 3. So does anybody have an idea how long, on average, this takes? Just guess out there. 10 years? Yeah, 10, 15, or in our case, greater than 1,000 years. So you can see on the left here, we show an early cave laboratory notebook drawing of a large mammal experiment showing that the mushroom is definitely a phenotypic hit for the ineffable. So imagine how much material was screened over that period of time. Stuff that didn't work at all, stuff that turned out to be toxic. All sorts of things are happening. But as you kind of head along and you start looking into like Sandoz era when Albert Hofmann actually synthesized psilocybin, we actually have a point where we can say we've got a lead. Okay, this is the lead, we're gonna move forward. And from there, we had all sorts of early safety and efficacy trials that went on very early in this research. But for a number of reasons, we can't use those other than the basis of evidence. We can't use those in our new drug application in the FDA. So Usona basically met with the FDA. Here's some Usona dates. We met with the FDA in 2017. By mid-2017, we'd established a chemistry laboratory, both in Wisconsin and in San Luis Obispo, California. And then we filed our new drug, our initial new drug application, investigational new drug application. And by just before 2019, we had our pharmaceutical grade psilocybin. After that, the FDA approved that we could proceed to phase 2. Now, this is a little bit different. Why did it take us so long to go from a chemistry lab in mid-2017, almost more than a year, to get pharmaceutical grade psilocybin? Well, this is because you have to think about the FDA was holding us to this requirement of commercial sponsor where they needed to really understand that we had safety and we had efficacy under control. In other words, they need to know the drug quality. And drug quality to the FDA is different than the drug quality to all of us. Okay? In the FDA, it's hard to explain what drug quality means, and I'm going to try and just walk you through this. To start with, let me show you what it doesn't mean. Okay. Playa mouth. Okay, so on this graph right here, on the vertical axis is 8 different batches of psilocybin. 6 of them are research batches made by people like Dave Nichols, Dan Cozy, Robert Carglow, Alex. A bunch of people have made batches like this. 2 of these are acceptable as GMP or pharmaceutical grade in a commercially sponsored. On the horizontal axis is purity. And so it's just from 0 to 100% pure. And if you look at the red bars, you will notice that whether it's a research batch or a GMP batch, they're all essentially 100% pure. They're all within 0.1%. And so the key to bring out of this, even though the blue bars might be a little bit different, that's just the impurity of water. So some of them have a little more water. All of these would occasion a mystical experience, right? But as the FDA, only 2 of them are acceptable. That's because in the eyes of the FDA, purity does not equal quality, all right? So what equals quality for the FDA? We're gonna back up a little bit on how we make these molecules to kind of get into that. So here's some chemical synthesis. Now, if you didn't take organic chemistry or you don't remember organic chemistry, 30-second review. Wherever you see an angle, there's a carbon. And if it's not a carbon, there's some element that's drawn there, an oxygen or a chlorine or a nitrogen or something like that. And so we kind of start in the upper left, and we start building on a side chain off this indole ring. We start building off a side chain. You can see where it's yellow is where the bond is forming. And then we get into something that looks like a tryptamine, but it's got these oxygens on it, and we reduce down those oxygens to something that's on the far upper right, which is psilocin, which is the active drug, as Alex had mentioned, the active drug for psilocybin. Now, psilocin isn't very stable, so we can't put it in a capsule and sell it or give it away. And so then after that, the phosphate is installed, and the phosphate has some protecting groups on it. And coincidentally, one of the protecting groups moves over the nitrogen that nobody really planned on, but it happens. And then finally, we use hydrogen to eliminate those protecting groups. The reason I throw this up here is because for a good chemist like Alex or Dave Nichols or any others, they can work through the anomalies of this chemistry. They can work through the fact that psilocin's not stable, that this benzyl compound in the lower left is not stable, that this zwitterion in the middle bottom is not soluble in pretty much anything. They kind of use those to their advantage, and they tease out the psilocybin. The FDA doesn't want that. They don't want talented chemists to be able to make the compound. They want anybody to be able to make the compound, okay? So first thing we did is we kind of redid the synthesis, because on the bottom, palladium on carbon, you might see it in there. The FDA hates heavy metals in their drugs. So that one we want to get out of there. All these compounds that are unstable or insoluble, we'd like to get out of there. So we redid the synthesis, and after about 6 months, found a new synthetic route that eliminated all of those detrimental aspects and made it a shorter synthesis. Also improved the yields by about 10-fold, so making it significantly less expensive. You people are the first people to hear of this new synthesis and to see it up there, and you'll see it published come out in the next quarter or so. So, what it is about this that gets back to FDA quality? Well, first, we're chemists and we like to distill structures. But the FDA really cares much less about the structures. They care about this diagram, which we'll say is the steampunk version of psilocybin synthesis. It's basically a flow diagram of all of the unit operations that occur for that last step. Reagents flowing in on the left, doing the reaction, followed by quenching, followed by filtering out solids and retriturating them and going all the way on through to the very end. And at the end, you get a drug. Well, the difference between research grade and GMP grade is this right here. The only control for research grade is you just look at the specifications of that drug. Is it pure enough? If it's pure enough, that's research grade. But to the FDA, quality is the control of the entire process. And so we start looking at other aspects. We control every reagent that goes into this by looking at the analytical view of the critical material attributes. We have documented recordings of the commissioning of the analytical equipment, the verification that that analytical equipment actually measures these material attributes. In addition, we have another layer of control, all of the engineering controls around each of these reactors, the temperatures, the stir rates, all of that. In addition, we have even more control where we're sipping out material out of each of those unit operations and analyzing them and documenting that analysis along the way. This is what GMP is. The entirety of the control of the whole process. This makes pharmaceutical grade psilocybin. And so in the end, what you have is basically these GMP reactors, and we can do one batch in about 6 weeks. We can make 1 kilo or 40,000 doses of psilocybin used for phase 2 and phase 3 trials. So next up is just, you've seen that graph already before. After this, we go into our phase 2B POC for efficacy, and then do a few more clinical trials, renal impairment, food effect. We do a few non-clinical trials in toxicity around liver enzymes and what are induced and what are inhibited. And then we continue on with a few tweaking on some of the synthesis. And that's what it takes to do the manufacturing of psilocybin for FDA.